Good afternoon. Thanks for stopping by. I'm going to be talking today about developments of disposables, typically for point-of-care applications. These are devices that would go into a small handheld or a benchtop instrument that would take the things that used to be done at the lab bench with pipette tips and laboratory personnel and put into a device and done automatically by the instrumentation. ALine has a unique platform that allows us to do rapid prototyping and design changes, as well as incorporating complex functionality such as valves, vents, pumps onto the disposable to allow us to do the sophisticated things that would be done on the benchtop. We've been in this field for 8 years now. The company started 8 years ago. And really our focus is to work with our clients to help usher in this future in human health care and stewardship of the planet by helping people to take the reagents and the know-how that they have for tests and tools and put those into a small footprint for disposable instrumentation and applications in the field. We find that even though they seem to be similar tests, there's always a unique challenge for each different kind of product that people want to develop, and we work very closely with our customers to help them solve those problems. Well, the drivers in this market, especially for the medical market, are compliance. That's one of the biggest health issues, is patients, even though they're taking a therapy, don't actually stick to the therapeutic regimen. And it would be nice if you had home-based tools that would allow the doctor to work with the patient to help monitor their health by home-based diagnostics. Of course, glucose monitors, as you know, is the most well-known and widely used. That's really the killer app in sample-to-answer devices that's on the market now. And in the future really is to take some of that same technology and make it more sophisticated and do more sophisticated things. The other things that are valuable for point-of-care testing are in the area of theranostics that really help to optimize the dosing. And we worked with a company called Osmotech, they're now Genmark, to develop a device that measures the patient's DNA to determine if they metabolize warfarin properly, and that's a blood thinner medication. Obviously, if you overdose the patient with a blood thinner, they are going to become internal bleeders. So if you're able to determine the patient's dosing level appropriately, then you support the proper dosing of therapeutic. Another important area in point of care or in where these microfluidic and small volume devices are making a big impact is in the development of cancer markers and cancer diagnostics, and this would be able to understand which patients should be taking which drug to treat their particular cancer. And also in general, there's a need to be able to support evidence-based medicine in order to have measurements of important metabolites or drugs that help to guide what the patient should be doing to improve their health. And that, of course, would help to reduce the cost of care because now we're moving into the preventative medicine. And so in all of those areas, microfluidics really can enable these kinds of applications. They offer compact, single-use products. The important part is tests that anyone could administer and to provide calibrated, often quantitative and consistent results, and that can be used in a point of care or in a distributed testing regime. Microfluidics would have needs in multiple markets. We've worked with customers to develop products for cell culture. This is an example of a fluidic card that flew in a satellite with a NASA project. This was the device that had a single inlet with multiple sample wells that connected to a satellite package that was no bigger than this and about that long, that automated and was allowed to do some cell culture in space to determine the effect of radiation and weightlessness on growing cells. The other application is doing molecular and immunodiagnostic tests. Many of the things that are done in the central laboratories are going to be shifted onto the point-of-care platform. Another important application is to be able to do multiplex and automated testing, particularly in life sciences, where you would like to, this is at the front end of the drug discovery process. And also this general field of doing sophisticated multi-step functional tests on a chip. So what we do is we provide expertise in the early engineering. Because of our rapid prototyping platform, we're able to shorten the product development cycle. Our laminate fabrication process doesn't require any tooling, so we can go from a design in AutoCAD to sample parts that have sophisticated functionality, valves and vents and pumps, in a matter of 3 to 5 days at costs that are significantly less than an injection molded tool. We also have noted that there's innovations in microfabrication technology that will drive down the costs for these disposables that have more sophistication, and we are helping to try to bring those pieces together. There's a lot of silos of expertise where some companies do injection molding, some companies do things in glass, others do things in plastic. We think it's going to be important to integrate and innovate in those areas and bring them together to try to meet the functionality as well as the price target. It's also important to think about design for manufacture from the very beginning. I think one of the things that's made microfluidics have a hard sell is that most of the designs and approaches to fabrication are very good in a research laboratory, but they don't translate into a commercial application. There's far too many risks of working at very small channels. So in most cases, if you want to have a product that's going to get to market and that your customer isn't going to be worried about being sued because of lack of performance, you want to stick to, in general, you want to stick to some pretty standard approaches to meet those objectives. And that's the way we try to work with our customers. We don't want it to be the sexiest looking device. We want it to be the one that meets the requirements for robustness and functionality. And so that would mean, meeting that price target and delivering the quality that's required. So what we see as important early in the product development is we develop a set of specifications, we develop a design that's going to meet that, and that design also has to work with the instrument platform that it's going to work with. And then we go through this build and test cycle rapidly and a number of times. So typically design, build, test, and modifications of both the instrument interface and the disposable happens many, many times. And the faster that we can deliver new prototypes to meet those test requirements, not only the better the product, but the faster we'll work with the customer to get to something that's very reliable. And we think that's an important part of what our platform offers that an injection molding doesn't offer. And so when we say this rapid prototyping platform, it's not just that we can deliver it fast, but we really rely on computer-aided designs to produce 3D objects without tooling. And those are really the important fundamental aspects of this technology. It's also amenable then also for high volume production. So the way the process works is we take a two-dimensional CAD design, we break the design into different layers, and then we specify the materials and the thicknesses based on the volumes and the functions that need to occur, and then develop an assembly process to facilitate volume production. We use a CO2 laser cutting. We can get to channel widths that are 125 microns with feature densities of about 0.75. The layers can be as thin as 12.5 microns up to about 3 millimeters. And as you can see in this cross-section here of this same cell culture card, that we can do stacks of up to about 13 layers with different thicknesses of materials to get the final device with the right volumes. We align, stack, and bond these using lamination techniques, and that allows us to get to 25 to 50 microns, 1 or 2 thousandths. Reproducibility on the alignment. The bonding adhesives are also 25 to 50 microns thick. As you can see here, we have porous membranes that are inserted into the back, into each side of this well. that allows the sample. In this case, there were E. coli that were grown inside of here. Those are non-adherent cells, so they were captured in that well. But it also, because of those membranes, they were like precision resistors in a circuit. It allowed a single inlet to feed a fresh glucose solution to all of those wells at the same time without having to have multiple inlets. So that was certainly an advantage for that project. So the nice thing about the polymer laminate technology is that we use a variety of commercially available biocompatible materials. They have smooth surfaces. We can easily get to a number of useful geometries with feature sizes to 125 microns and incorporate valves and vents, again, sticking with this lamination process. And the manufacturing processes that we're developing really build on the things that have been done early on with glucose strip tests with lateral flow devices, where you do the combinations of adding reagents or thick film printing and then adding layers on using the same manufacturing processes that everyone in the medical device industry is very familiar with. The other nice thing about the technology, because we do have bonding surfaces on the bottom of the device, you can interface them readily to sensors or manifolds. Because of our experience in this area for 8 years now, we've developed standardized subassemblies and automated processes so that we can work with customers to direct the design, to do the complexity in certain components, so that we can do better in-process QC of those processes and lower the cost of manufacture. Because we don't change the process, we just change the arrangement of things in 2D. Another important part of developing these point-of-care devices is really to understand the interface of the fluidics to the instrument, and working closely with an instrument developer to do this is key to getting the final result. It's not just the design in the disposable, it's how it works with the instrument. In general, you'd like to be able to have the instrument easily pneumatically or with vacuum or a combination of the two to support metering, mixing, and pumping on the device. Temperature control is important, particularly for PCR. I know a lot of people fail in getting a proper interface between their plastic device to their heater for PCR, and they call us and say, "God, what's the melt temperature on this plastic?" Well, it's 110 C. Well, it's melting. Well, you're not reading the temperature correctly. So you really have to work with customers to help them understand how to get these things up and going to get on the breadboard instrument. I think one of the main issues people have had with microfluidic technology is the cost of manufacture. When you start putting in the sophistication of pumps and valves and vents and so on, you're talking about cost of manufacture with injection molding plus post-processing plus reagent addition. You're bumping up into $13, $20 per each, and you can't manufacture a device and make money if that's the cost of manufacture. So the target really is to have cost below $5 a piece. To get that sophistication, that's going to probably mean a multiplex test. It's going to mean onboard reagents so that you can deliver the functionality at a right price. But because of this is a very multidisciplinary technology, so in order to do it successfully, it's nice to work with someone who understands the chemistry and the biology, the materials, and the different fabrication methods, and then the utility of different functionality to support putting something that was done in a microtiter plate onto a disposable. So there's always these issues of sample processing, what kinds of detection methods, and so on. And this platform has the kind of flexibility that allows you to explore any number of different detection technologies. We've interfaced for surface plasma and resonance, chemiluminescence, fluorescence, as well as electrochemistry, electrochemical detection. So we feel that as a company, what we want to do is to support this value chain from getting from the early R&D to commercialization, help get from these initial designs through these prototypes and then into production. And we've successfully done that with Osmotech, supporting them from the early rapid prototyping when there were a lot of design changes, all the way through clinical trials and then to product launch. And we supported them in production for the first year and a half after the product was commercialized. We also were awarded as a supplier to them. They won the Medical Design Excellence Award in 2009, and we were honored as a supplier. So what we feel we need to do is to work with our customers to reduce the complexity, shorten the development time, and lower the risk, which all of these things feed into enhanced investor confidence, because usually we work with companies that are getting investment to support their development. Right now the market is very fragmented. You've got companies developing products and they're looking for different technology providers, instrument developers, reagent providers, and we want to try to bring together a network of vendors to help support doing that for our customer. So we have developing capabilities in addition to the lamination. We're now driving towards being able to broaden the technology platform. So we are now getting into doing the embossing or compression molded parts, broadening our bonding capabilities by being able to do heat, pressure, and solvent assisted when it's appropriate, and also to integrate surface modifications to improve the flow of fluids in the device, as well as being able to do reagent deposition and thick film deposition for electrodes. We're also working in partnership now. We're teaming with Pango Medical to try to bring to market OEM systems that have either electrochemistry detection or the optical detection. And these are systems that are about 70% ready for commercialization. All they need is the appropriate reagents put in the disposable and the final interface and development, and they could be privately labeled point-of-care systems. So ALine's been working to develop strategic partnerships to integrate this value chain and integrate and automate the fabrication technologies to drive down the costs, developing these pilot scale capabilities. This really helps to lower the cost to our customer because they're not going out and looking to develop manufacturing, high volume manufacturing, and output of all the tooling costs. So we can work with them to get to market without a huge investment in capital to get there. And we have developing our markets in, of course, in the U.S., but also in Europe. So our capabilities include the design and engineering, the development of hybrid devices that include valves and electrodes. We have a couple of products for spectroscopy, both a microvolume cuvette for fluorescence and transmission experiments. We can create devices that already have built-in modularity for valves and pumps. And we can do this rapid prototyping. We have an FDA-compliant quality system because we were doing manufacture for Class 2 medical device. So this is, I'll just use this as the best example of how our platform really works in a real product. This is the Genmark's XT8 EXT platform for warfarin measurement. This is the printed circuit board that has the gold detection pads here. It runs along what they call the racetrack, and the laminate that you see here has also has channels in it, but it also has 2 bridge valves and a pump here that when you actuate through here onto this diaphragm causes the fluid in this area to recirculate, and that of course increased the kinetics of binding of the DNA probes to the gold pads on here. So this is an example of a multiplexed molecular detection technology that really took advantage of the laminate to be able to make a complete device by just using the pressure-sensitive adhesive to bond the printed circuit board with the injection molded component and put it into this complete package for the disposable. So the injection molded part here really is the interface to the outside world, and then the experiment happens there, the test. And then again, here's the project we work on with NASA for doing cell culture in space. This is the satellite package that has the electronics here. This is a little area that contains a fluid pouch that has the glucose, buffer with the glucose to feed the cells. And you can see two little tubes come into there, into the end of this card, through these ports to feed all 10 of these wells. These two are controls for the optics. This is the optical rail here. They did both fluorescence and transmission measurements. And they looked at the expression of green fluorescent protein in this engineered E. coli. So the way that this worked was to displace the stasis media with the feed media. Once it was launched into space, the fluid came in through this port and then would fill through the membrane and come up and exit out through here. And again, because the membranes all have the same surface area and the same porosity, we were able to use them, just one inlet line, and the fluid was displaced in all 10 of those wells at the same rate. That was an important function for that card. So our competitive advantage is that we engineer for production. We have a flexible platform that doesn't require tooling, that allows us to do the rapid prototyping, but at the same time can get to volume manufacture using all of the standard manufacturing processes that are used in the device industry already. And I think I'll just end it with that. I think I've pretty much said all of this already. Yeah. So we've worked with a number of customers in industry, government. We've had one project with we were a subcontractor on a government program for doing fieldable tests of bad bugs. So this was a true sample to answer system where you take a swab, you put it up in fluid, and then everything went into the cartridge: cell lysis, PCR amplification, purification, and then detection was all launched on a card that was built using our platform. And we're producing that now for that project, about 100 a month on those. So I think I'll end it there. We still have 10 minutes. Thanks. Are there any questions?
Do you modularize your technologies so that they can be used on different multiple platforms and for different applications?
You mean modularize in terms of whether it works in one kind of instrument or another?
Yeah.
Well, no, those are typically, so the interfaces would be custom to the instrument platform and the way that the customer wants to sell the package, how they want it to interface with the user.
But is there a way to standardize different modules so that they can be used across different multiple platforms?
Well, I mean, certainly it's very easy to standardize to the automation platform, either 9 millimeter or 4.5 millimeter centers for robotics. So the same thing could apply for interfaces to an instrument, where you could put everything on a standard spacing at the edge of the card. Yeah. Thanks. Well, thank you for your attention.